Now we begin to talk about the histology of the small intestine. Again, the wall of the small intestine has the same general layout seen in the rest of the gastrointestinal tract. Namely, it is formed of mucosa, and the mucosa is formed of epithelium, lamina propria, and we have muscularis mucosae, this layer here, which separates the mucosa from the second layer, which is the submucosa. So this is the submucosa. Then we have the third layer, which is the muscularis externa. And finally, the outermost layer, which could be either a serosa or an adventitia, depending on whether the organ is covered by peritoneum or not. Now, the parts of the small intestine that are covered by peritoneum, the outermost layer is a serosa formed of connective tissue and epithelium. If the part of the small intestine is not covered by peritoneum, then the outermost layer is formed of only connective tissue adventitia. Now, permanent circular folds of mucosa and submucosa are present in the wall of the small intestine, and these are called plicae circularis. So imagine, first of all, this is the longitudinal axis of the small intestine, and this is the transverse axis of the small intestine. Now we have folds running along the transverse axis circular, like these, all these, and these are the plicae circularis. They start in the duodenum. They are most numerous in the jejunum, but they are few or even absent in the ileum. So if you look at this first image here, this is the proximal jejunum. Notice how we have many folds, many plicae. All these are plicae. Whereas in the proximal ileum, we have just a few, and in the terminal ileum, we have none. No plicae circularis, no folds. Now, these folds increase the surface area of absorption and slow the passage of food through the intestine, allowing more time for the intestine to absorb nutrients. Now, since these are circular folds, food has to zigzag between them in order to get from one point to another. This will slow the passage of food through the intestine, giving it more time to absorb nutrients. And this is why these are most numerous in the jejunum, because most of absorption takes place in the jejunum. And this is why in the terminal ileum it is absent, because by the time food reaches this part of the small intestine, the terminal ileum, the absorption process has been almost completed and no folds are required. Let's start with the first layer, the mucosa. Mucosa, as we have said, is formed of epithelium, lamina propria, and muscularis mucosae. The epithelium and lamina propria, this is important, the epithelium lamina propria of the small intestine project into the lumen to form villi. These increase the surface area of absorption. Notice how this is the epithelium. It projects into the lumen, and with it also we have the lamina propria, also with the epithelium. These structures are called villi, the projection of the epithelium and the lamina propria. Between the villi, we have simple tubular glands. These open between the villi, and these glands are called the intestinal glands, intestinal crypts, or the crypts of Lieberkühn, an old name, crypts of Lieberkühn, the intestinal glands or intestinal crypts. So what are the cells of the villi? What cells do we find in the intestinal villi? We have two types of cells, enterocytes and goblet cells. Enterocytes are tall columnar cells. These are the cells that form the simple columnar epithelium that lines the small intestine. So enterocytes are tall columnar cells, and of course they have an oval nucleus which is usually located in the basal half of the cell. The apical surface of the cell is characterized by the presence of what we call brush border, which is actually formed of numerous microvilli that contain the digestive enzymes. So here is a transmission electron microscope image of the columnar cells. This is the cell, and here we have the surface of the cell. Notice how on the surface we have numerous closely packed microvilli. And these microvilli form what we call the brush border or the striated border of the small intestine. Now, these microvilli contain the digestive enzymes, but in addition, these microvilli increases the surface area of absorption. Now, so far we talked about the presence of folds. We have folds in the small intestine. We have villi in the small intestine, and we have microvilli. So remember the folds, the plica circularis, are formed of mucosa and submucosa, villi, which are projections of epithelium and lamina propria, and finally we have microvilli on the surface of enterocytes. All these structures help to increase the surface area of absorption for the small intestine. And actually, these structures greatly increases the surface area of absorption of the small intestine. The other cell is goblet cells. These are mucus-secreting cells. They are found between the epithelial cells. They are less in the duodenum and they are more in the ileum, which we are going to talk about later. The apical part of the cell is distended with mucinogene granules. These are mucus-secreting cells, so the vesicles or the granules found in the apical part, these vesicles contain mucus. Golgi apparatus forms a wide cup just below the granules. All this part is occupied by Golgi apparatus. The stem-shaped, notice the stem-shaped, this part, basal region of the cell contains the nucleus, numerous of endoplasmic reticula, and mitochondria. Finally, microvilli are restricted to a thin rim of cytoplasm that surrounds the apical part. So it is just on the rim of the apical part and does not cover the entire apical surface. So notice how the basal part of the cell is stem-shaped, whereas the apical part is wide, and this is why it is named goblet cells, because its shape is like a goblet. It is shaped like a goblet. So the epithelium that is found in the villi is formed of two types of cells, enterocytes and goblet cells. What about the core of the villus? What do we find in the middle here? This region, the core of the villus, of course, is filled with the connective tissue of the lamina propria, and within it we find an arteriole, a venule, and an lymphatic vessel that are connected to submucosal vascular plexuses. Now remember, of course, that absorption takes place here, so the absorbed nutrients pass into the vessels. Of course, you have to remember that fat is absorbed into the lymphatic vessel, and this is why it is whitish in color, and this is why lymphatic vessel found here is called the lacteal. lacteal معناها حليبي لأنه يكون مليء بالمادة الدهنية الممتصة. In addition, within the villi, we find smooth muscle fibers that are derived from the Muscularis mucosae. From the muscularis mucosa arise several smooth muscle fibers. These are important for the rhythmic movement of the villi. These smooth muscle fibers help the villi move around. So, what are the cells that are found in the intestinal glands or in the intestinal crypts? Here we have enterocytes and goblet cells, the same cells that we talked about earlier. We have enteroendocrine cells, the endocrine cells of the small intestine. They secrete various hormones like cholecystokinin, secretin, motilin, and others. We have stem cells. These replace all the other cells of the glands and the villi. Now, enterocytes produced from these stem cells migrate from the crypt to the tip of the villus where they die. The stem cells are found in the crypt. They form enterocytes. The enterocytes will start to migrate upwards or superficially. They leave the crypt and they pass into the villi. They continue to migrate until they reach the tip of the villus where they die. This cell turnover takes about 5 days. So every 5 days we have the formation of new enterocytes. Paneth cells, these produce various substances into the lumen of the intestine for the non-specific resistance against organisms. An important and unique cell found in the intestine are the microfold cells, also called the M cells, M short for microfold. These are present in the ileum, overlying Peyer's patches. Peyer's patches is a collection of lymphoid tissue found in the submucosa of the ileum. So the epithelium superior to it, superficial to the Peyer's patches, contain these microfold cells. So in this image here we have enterocytes. These are enterocytes. This is another enterocyte. And these are the M cells. These are the M cells. Now, the apical surface of the M cells has several microfolds. This is why it is called microfold cells. Notice the upper surface is folded, microfolds. But the characteristic feature of this cell, a unique feature of the cell, is that the basal surface has a membrane invagination. Notice how the cell membrane in the basal region invaginates the cell, forming a subcellular pocket, a pocket just below, just inside the cell. This pocket contains lymphocytes and macrophages. So we have cells found in the pocket of the microfold cells, macrophages and lymphocytes. So why is this arrangement? And what is the function of these microfold cells? Function of these cells is the non-specific uptake of antigens from the intestinal lumen. So if there is an antigen in the intestinal lumen, for example, these antigens, when they reach near the microfold cells, they are taken up by these cells, they are engulfed by these cells, they are transported, these antigens are transported inside the cells, and then they are released, these antigens are released into the subcellular pocket. So from the lumen into the cytoplasm of the microfold cells, and then from the cytoplasm into the subcellular pocket. What do we find here? We have macrophages and lymphocytes. These will take up these antigens and process them, and this will lead to activation of the immune system. So the microfold cells act as a bridge between the antigens in the lumen and the lymphocytes and macrophages and the immune system in the connective tissue below. Now, if you look at this image here, these are the villi, and here we have the crypts, the intestinal crypts. If we take a section through a villus, we'll have a cross-section, a circular cross-section. The cells are on the outside, and in the middle, we have the connective tissue of the lamina propria. The core of the villus, as we have said, is filled with the connective tissue of the lamina propria. Notice how the lumen here of the intestine is outside. The lumen is outside. Therefore, the secretions of the cells are to the outside. Secretions are to the outside. Of course, absorption is in the opposite direction. Absorption is from the lumen into the connective tissue, but the secretion is from the cell to the outside, into the lumen. Now, if we take a cross-section through the crypt, cross-section through the intestinal crypt, we'll also have a circular cross-section, but the lumen is on the inside. The lumen is on the inside. So the secretion of the various cells, the cells that we have talked about, enterocytes, goblet cells, Paneth cells, all these secretes to the inside of the crypt because the lumen is on the inside. However, the enteroendocrine cells, remember, they are endocrine cells. They release hormones. The hormones pass into blood vessels. The blood vessels are found in the connective tissue of the lamina propria. For the intestinal crypts, the connective tissue is outside, so the lamina propria is to the outside. So here, the connective tissue of the lamina propria is outside the crypt. And therefore, the neuroendocrine cells release their secretion to the outside. The other cells release their secretions to the inside, into the lumen. So what about the other layers? We have been talking about the epithelium. What about the other layers? The other layers of the mucosa is the submucosa. The submucosa of the duodenum has duodenal or Brunner glands of the duodenum. These are mucus glands. They secrete mucus. This mucus is important to lubricate the intestinal wall, to neutralize the gastric acid, the food coming from the stomach into the duodenum, in case it is still a little bit acidic. This will help to neutralize this acid. It provides also the optimal pH for the action of the enzymes. So in this section here, notice these are the villi. This is the submucosa. These are all the mucus glands, Brunner glands. Notice they are colorless because these are mucus glands. Now, this is why we said that goblet cells in the epithelium are less in the duodenum, because we have numerous mucus-secreting Brunner glands in the submucosa of the duodenum. But we also said that goblet cells are more in the ileum. Now, why is that? Now, most of the absorption takes place in the duodenum, and by the time food reaches the ileum, absorption is almost complete, and what remains is the undigested food material. And this is usually harder. This is relatively harder. So by the time food reaches the ileum, it is hard. It is a hard substance, and therefore we require some protection. This is why we require mucus secretion in the ileum to lubricate the area, to prevent or to protect the ileum from the hard food particles. This is why we have more goblet cells in the ileum. We are going to see a very similar situation in the large intestine. Also, in the submucosa of the ileum, we have a collection of lymphoid tissue called Peyer's patches. This is a section through the ileum, and all these are Peyer's patches. Of course, it doesn't occur throughout the ileum, but it occurs in a certain region, a specific region of the ileum, one side of the wall. And notice these large lymphoid tissue which are called Peyer's patches. Of course, these play an important immune role. And also, we must remember that within the submucosa, we have the submucosal plexus. The muscularis externa is formed of only two layers, an outer longitudinal and an inner circular layer, between which we have, of course, the myenteric plexus. The outermost layer of the small intestine could be either a serosa or an adventitia. If the organ is intraperitoneal, covered by peritoneum, the outer layer is serosa, formed of connective tissue and epithelium, and this serosa is continuous with the mesentery. However, if the part of the small intestine is retroperitoneal, for example, parts of the duodenum, the outer layer is a connective tissue adventitia. Last topic is the histology of the large intestine. As in the other parts of the gastrointestinal tract, the wall of the large intestine is formed of mucosa, submucosa, muscularis externa, and serosa or adventitia. So we start with the mucosa. The large intestine is lined by simple columnar epithelium, and this simple columnar epithelium passes into the lamina propria to form tubular intestinal glands. So this one, the epithelium here will pass into the lamina to form the glands. And the epithelium of the large intestine is formed of three types of cells: colonocytes, goblet cells, and stem cells. Goblet cells, the same as in the other parts of the gastrointestinal tract, but these become more numerous as we go distally along the large intestine, because as we move along the large intestine, the food particles become harder and therefore we require more lubrication, and this is done by the secretion of mucus from goblet cells. Stem cells, these are located in the bottom third of each gland, and as usual, these are undifferentiated, highly mitotic cells that divide and differentiate to form all the other cells of the large intestinal epithelium. So here we have, first of all, we have the mucosa formed of epithelium. Notice the epithelium passes down into the lamina propria to form the glands. And then we have lamina propria, connective tissue. We have muscularis externa. Under the mucosa, we have the submucosa formed of connective tissue. Finally, we have the muscularis externa. Now, the outermost layer of the large intestine could be either a serosa or an adventitia, depending on whether the part of the large intestine is covered by peritoneum or not. And in this histological image, this is the epithelium. We can see most of the cells, these are the glands. Most of the cells are white or colorless. These are goblet cells. So as you can see, goblet cells is very numerous here, very numerous. Most of the cells are actually goblet. And the more we go along the large intestine, the more abundant goblet cells become. Colonocytes, the special cells of the large intestine, these are columnar cells. So since we say columnar, this means that they have oval nucleus situated in the lower part of the cell. They have short microvilli at the apical surface. So here is the columnar cell, and this is the nucleus. These are the microvilli in the apical part of the cell. Now, the microvilli here are short and dispersed, so they are not numerous, not closely packed with each other like in the small intestine, so there is no brush border here. Now, a very important feature here is the presence of large intercellular spaces between the cells. These are the large intercellular spaces between the cells. And this is a characteristic feature of the cells whose function is the absorption of water. Cells that perform absorption of water have this characteristic feature, the presence of large intercellular spaces between the cells. The lamina propria of the large intestine has the same features as in the other areas. However, it is rich in lymphatic nodules that extend into the submucosa, large lymphatic nodules that extend into the submucosa. And of course, this is due to the large bacterial content of the large bowel, and this is even more prominently seen in the appendix. Muscularis mucosae, the same as in the other parts, is a simple layer of smooth muscle cells. The muscularis externa has some special features in the large intestine. It is formed of two layers, inner circular and outer longitudinal, just like in the small intestine. However, the outer longitudinal layer has some special arrangement. In the colon, remember, the large intestine is formed of the cecum, colon, the rectum, and finally anal canal. So in the colon, all this, the ascending, transverse, descending, and sigmoid colons, the outer longitudinal muscle layer aggregate into three bundles called taeniae coli. Three bundles called taeniae coli. Now, so they are not continuous, they do not form a continuous ring around the organ, but they are present as in the form of bundles at specific regions. And the taeniae coli are shorter than the large intestine, so this will lead to puckering of the colon into large sacs called haustra. Notice how we have these expanded sac-like structures here. These are the haustra of the large intestine, and these occur because the muscle is shorter than the actual length of the colon. Serosa or adventitia. The intraperitoneal parts of the large intestine are covered by serosa. So if the organ is entirely found inside the peritoneum, so it is entirely covered by serosa. The retroperitoneal part, we have some parts covered by adventitia. Now the serosa of the colon form several pendulous fat-filled sacs called appendices biploici, or the omental appendices. Notice these various appendages here. These are the appendices epiploici. These are derived from the serosa. The last part of the large intestine is the anal canal. Now, of course, again, the same general layout, the same layers. The anal canal shows several longitudinal folds of mucosa and submucosa called the anal columns. The anal columns. The rectum shows three prominent transverse rectal folds caused by enlargement of the muscle layer of the wall. Okay, so the folds, the rectum is different. It is due to the enlargement of the muscle layer. The rest of the large intestine shows only few transverse semilunar folds. So now, folds of mucosa and submucosa has different names in different regions. For example, in the stomach, it is called rugi. In the small intestine, it is called plica circularis. In the anal canal, it is called the anal columns. These are all folds of mucosa and submucosa. In the lamina propria and the submucosa of the anal columns, we have sinuses of the anal venous plexuses. When these sinuses are enlarged, they are called hemorrhoids. So hemorrhoids are enlarged venous plexuses found in the anal columns. Now, in the lower part of the anal canal, the circular muscle layer is thickened to form the internal anal sphincter. Again, the muscularis of the anal canal has an inner circular and outer longitudinal layer. In the lower part of the anal canal, the inner circular layer becomes thickened to form the internal anal sphincter. And of course, this is involuntary sphincter. Outside it, outside this sphincter, we have the external anal sphincter. This is formed of skeletal muscles and therefore it is voluntary. What about the epithelium? If we connect the lower borders of the anal columns, we get a line called the pectinate or dentate line. Pectinate or dentate line. At this line, the simple columnar epithelium of the rectum will change into stratified squamous non-keratinized epithelium. So here we have rectum. Here this is the rectum. And then the anal canal will start. And then at a specific region, at this part, of course the anal canal has already began. But at this line, this is called the pectinate or dentate line. The epithelium, here it was simple columnar. It will suddenly change into stratified squamous, but it is non-keratinized epithelium. Stratified squamous non-keratinized epithelium. Now further inferiorly, approximately at the level of the interval between the internal and external anal sphincters, at a line visible in the living person, which is called the white line, the epithelium becomes stratified squamous keratinized. So if we look at this image here we have the rectum lined by simple columnar epithelium. The upper part initially of the anal canal, the initial part of the anal canal, lined by simple columnar epithelium. These are the columns. These are the columns, anal columns. If we connect their lower border, now notice how the lower border of the anal column is not a straight line, but it is undulating. This is why it's called dentate line. متموج يشبه الأسنان فيسموه dentate line. At this line, the simple columnar will change into stratified. So in this region here, we have stratified squamous, but it is non-keratinized epithelium. Now here we have the internal anal sphincter. Outside it, we have the external anal sphincter. There is a certain point, a period, there is an interval between them, this region here, and this is visible and can be felt, and this is called the white line of Hilton. And at this line, the stratified squamous non-keratinized epithelium of the anal canal will become stratified squamous keratinized epithelium, just like that of the skin. And by this we have ended this lecture of the histology of the stomach and the small and large intestines. Thank you and good luck.